Thank you for joining. I would like to begin by congratulating the Molecular Pathology Association of India for reaching this momentous milestone. This is the 10th annual conference. It's a big milestone, and I'm sure there are many more to come. So many congratulations and the best wishes. I would like to thank Dr. Rahman, Dr. Mishra, and the organization committee for allowing me to join this conference and present some of the work that we have been doing. I'd like to present the evaluation of NGS in clinical practice and share our experience at MD Anderson Cancer Center at Dr. Mishra's request. So that's going to be the presentation that I'll be sharing today. We will go over the journey that we had over the years. We have been performing NGS testing for more than 10 years now. And so I'd like to review a little bit of what we went through in terms of the initial implementation, and then follow the journey in terms of what we did with the NGS panels that we designed, whatever the additional applications that we are developing, as well as some of the important quality and patient safety issue. There is a lot to share, but within the confines of the allocated time, hopefully I can provide some high-level glimpse as well as the lessons that we learned during this journey. So this sigmoidal curve is not unfamiliar to anybody in the audience here. This curve, whenever we see it, it immediately reminds us of the different phases of PCR. However, today I'm sharing this curve here not to show the PCR. However, I'm sharing this here to remind us that the topic of the discussion today, which is the next generation sequencing, is actually a technology. And this is showing what we call a technology curve. And hopefully I can show some parallels in terms of how NGS has really arrived and has really sort of found a foothold in the clinical molecular diagnostic community. So if you expand this is what a typical technology curve looks like. When a new technology emerges, there is an emerging phase, then it goes through what we call a growth phase, maturity phase, and then saturation. And because this is a cycle, it gives way to a new paradigm, a new technology, and that technology goes through the same phases again. And just how the new technology has replaced a previous paradigm or a technology, something new will replace that. Right now, I consider that we are somewhere in the growth and maturity phase, but definitely not in the saturation phase as it relates to the next generation sequencing. So I'm like to draw some parallels here and then show our journey through different phases, and hopefully we can follow along from emerging to the growth to the maturity phase in the next few slides. Initial phase or the emerging phase for us really started around 2010-2011 when the first benchtop NGS sequencers arrived, and these were the INPGM and the MySeq sequencers from two different manufacturers. At the same time, what was happening in the diagnostic community was there was a explosion of predictive biomarkers on the solid tumor side, as well as there was a lot of novel discoveries coming in the hematologic malignancy side, which both meant that a typical molecular diagnostics laboratory had to develop a number of these different assays and different biomarker platforms to be able to provide a comprehensive profiling. And right around that time, these benchtop sequencers came along and really provided us a very useful tool for molecular diagnostics community. Around the same time, or at the same time, I would like to say that the adoption of such an innovative technology requires a teamwork, a comprehensive approach to this type of a new platform, and really an entire ecosystem where we come up with new processes. We create a climate to allow this new technology to come in. We train people, we starting from the leadership all the way down to the people who carry out the day-to-day function, and also accountability and oversight. At the same time, we create policies and procedures. So these are all topics in themselves of longer talks and discussions. But I just wanted to recognize the fact that implementation of NGS as a technology was really in this framework where everything aligned, and we were able to implement this very powerful and yet really novel technology in the clinical diagnostics, where for the very first time, instead of running one sample per well or running one sample per lane, we were now intentionally pulling multiple samples. And that can be scary, that can be nerve-wracking, as we can imagine. So, and that's one reason, or one of the many reasons, why we required a detailed framework. So what I'm going to be describing is actually part of this larger framework, and I'll be sharing some important bits and pieces from that journey so that we can understand different elements of this type of a ecosystem that needs to be created to allow a successful integration of a technology such as next generation sequencing. So on the left hand side, again, just going back to the beginning of the journey, what you are seeing there is what our molecular diagnostics laboratory, which is focused primarily on oncologic testing, so cancer, we maintained a variety of different platforms. Some of these we still do. We're actively working on updating them or replacing them with more efficient approaches. But you can see how a variety of these different platforms and different biomarkers being run on different platforms can create so many logistic challenges for the lab to maintain the platform, to split the sample, which can be very limited many times across these many different platforms, to be able to provide the information. And at the same time, from the clinical perspective, they may not get all of the information at the same time, thereby limiting their ability to provide the timely assessment and treatment decisions. So, but that was the only available platform. Those are the available platforms. And towards the late 2010, we had started performing limited amounts of sequenome, which allowed us limited amounts of multiplexing. That was the first entry into the multiplexing. But when the next generation sequencing arrived with all the benefits which are listed here, less DNA, consolidated workflow, multiplexing, and so on and so forth, it really took over from many of these other platforms, and it's almost become a Swiss Army knife of a molecular lab for us, where we were able to quickly consolidate a lot of these platforms onto a single next generation sequencing platform. I would like to focus more on the important lessons that we learned from this, so that we can share these examples. There has been a lot of literature now and a lot of knowledge in the community in general about how these platforms work and how these platforms can be validated. So I would like to share what we did in particular with regards to the initial approach. So we initially started out with the hotspot commercially available panels, which are again by two manufacturers, prominent manufacturers at that time. And this is the so-called AmpliSeq or TrueSeq based panels, which are range from 46 to 50 genes, with a lot of overlap, as you can see here, with maybe some content which is part of one panel but not the other. But largely speaking, there is a lot of overlap except for these two biomarkers, GNAQ and GNA11. So this did provide a very useful entry in a manner where we could understand the scope of this technology. This being a hotspot, the footprint was smaller, but we knew a lot of these hotspots, what to expect from these different genomic regions. And at the same time, these other genes which are included, and these are some of them were emerging genes, they are also part of the panel, allowed us to expand the genotyping beyond the conventional genes only. So this was really useful experience where we established a lot of workflows, a lot of clinical tools, and important quality control measures. Just to give you an example of ensuring that this technology results into safe environment and a safe platform, we did develop close to 162 new Sanger assays matching every single one of these amplicons, which is shown here are covered on this panel. So again, it took a lot of effort for us to ensure that this new technology that we're embracing is actually safe as well. Showing some examples of utilization after this test was utilized for a while. What we did note that the yield from this type of a generic off-the-shelf panel was limited. You can see the genes in biomarkers crossed out and in red are the ones that really did not show significant rate of mutations, and the remaining the ones that we knew are hotspot, they continued to show a good mutation rate, meaning good clinical utility. And this really also was an important message and the lesson in terms of the next phase of our evolution. At the same time, what we had noted is that there are some important gaps in some of the commercially available panel. For example, when you look at this panel here, this is the AKT1, an amplicon based panel. On the top is the original coverage, where instead of codons, amino acid codon 17 being covered, somehow the coverage was placed on codon 49, perhaps because of the C.49 designation. When we noted this, we went back to the manufacturer, they immediately corrected, and we got the coverage back on the region that was needed. Some other examples of this optimization phase that was needed on top of the commercial availability is an example in a bidirectional sequencing coverage. The cycle, if you went with 2 by 150 approach in forward and reverse direction, just because of the length of the amplicon, a typical hotspot, in this case a codon 617 in JAK2, was only covered in one direction. So we were able to change or shorten the forward sequencing cycles and increase the number of reverse sequencing cycle so that the hotspot was covered bidirectionally. So this type of optimization was needed to ensure that every target was adequately covered and we were getting the maximum benefit out of this very powerful platform. And that really led us into the next phase, which is the growth phase. So having found the use of this technology in the growth phase was defined essentially by continued expansion in the solid tumors, a lot of the histology agnostic clinical trials such as NCI match trial, TAPUR trials, and others coming in, where biomarkers were essentially directly driving treatment decisions without the histologic considerations. And also within the histologic context, a lot of expansion for molecular biomarker testing was happening across the different guidelines and standard practice. On the hematologic side, the WHO classification was revised in 2017. It introduced many new categories, which are again were biomarker based. New targeted therapies started to come out, which again led to increased demands. And during that time, or during this growth phase, I would say 2015 onwards, so the second 5-year block, we were seeing also on the NGS side, there was improved sequencing power. We were able to make the libraries more efficiently. So overall, the processes were much more robust, and we were able to accommodate this growth in demand, both in content as well as in the application during this phase. So during this phase, what we had done is we tried to move away from the off-the-shelf panels and try to come out with a custom design. And there are lots of C's here which are pointing to the custom design of the panel and the advantages. So obviously with the custom panel, you can control the content. You can come up with the appropriate coverage across the regions which are important for genotyping. We can identify the appropriate chemistry, appropriate clinical application, as well as it can also provide a unique competitive advantage if you are in such an area. And obviously it will give you a lot more cost efficiency since your target content is high yield and you're not wasting sequencing real estate on genomic content that is not high yield. And they can be compliance ready. And so there were lots of advantages, and keeping that in mind, what we had done is across our portfolio of hematologic malignancies as well as solid tumors, we introduced several new panels which were all had a custom design hand-picked content that would represent variety of different genomic targets as shown here. This is our 81 gene leukemia panel. We call it N leukemia SA1, which is primarily for bone marrow based hematologic neoplasms, and the content here does reflect that. So this is the type of considerations that went into these panel designs. They would also reflect the clinical application. So as I mentioned, one of the key drivers was WHO classification, novel targets for germline predisposition such as DDX41 and so and so forth. And also, since more and more signature-based prognostic profiling were being performed, this panel did allow that comprehensive assessment, and the content was high yield and still comprehensive enough that it continues to serve our purpose even till date, and very small amount of content needs to be added to this. However, at the same time, while we were able to deliver this comprehensive content, the therapeutic field kept moving along. And this is an example of of our IDH1 and IDH2 assays, for which fortunately there are targeted therapies available, but they would move these biomarkers from being purely prognostic in nature in the earlier phase to actually predictive in nature and in the later phase. And when that does happen, this information is required on a rather urgent basis so that the patient care decisions can be made. So as you can see, earlier on the goal was to provide the coverage and improve the lab efficiency, but later on that quickly changed to turnaround time optimization. And as they say, necessity is the mother of invention. It did force us to come up with our own unique proprietary platform, which is published here in 2019 JMD paper. We call it UrgentSeq, Ultra Rapid Review of Genomic Targets, where we are essentially capturing the live sequencing data from a live sequencing run when it passes a certain cycle and performing genotype calls on that unidirectional sequencing data and providing useful information for diagnostic markers such as JAK2, MEPL, calreticulin, or predictive markers such as IDH1, IDH2. We've recently also added P53 to this list. So this really provides us a very useful approach without adding much cost to our sequencing cost and effort. By providing a rapid turnaround, we can typically turn this around within 48 to 72 hours. This is an active process. It requires active pathologist involvement, but we are able to make significant contribution to patient care by being able to develop this innovation. At the same time, on the lymphoma, a lot of new biomarkers again were being discovered, new clinical trials, new therapies emerging. However, the lymphoma presented a unique challenge in the sense that the lymphoma samples can travel in a variety of different matrix. So it can be in suspension from an FNA, or it can be on the cytology smears. They can be on a core biopsy, or can be in an excision. So as you can imagine, trying to come up with a systematic approach where these biomarker orders are placed in the EHR in an appropriate manner, the sample is collected, the sample is transported, and eventually the sample gets tested in an appropriate manner. This does require a large number of team members, and hence the title here, the power of teamwork and tenacity. We were able to work through lots of challenges, and I'm pleased to announce that for last several years we've been routinely testing the lymphoma using our custom design, again 162 gene panel, which is what we call an lymphoma panel, and it does have coverage for a large number of non-Hodgkin lymphoma as shown here. Again, implementation of something like this can be very complex. So this figure here just shows that when we started at a certain time point, and in this left hand side we had different sample types and different tumor types. There are many blocks in red, meaning that we did not have a solution for those, but over time we kept working through them, and then eventually we are now able to offer this testing for all sample types, for all lymphoma types. Again, the point here being that this can be incremental. This may not just appear in green all of a sudden, and there will be a lot of work that needs to be performed and work through challenges to make it happen. Around the same time, there is also some additional developments happening now on the solid tumor side, where the liquid biopsy is emerging on the scene and really provided a very rapid turnaround for patients with solid tumors, and probably the first time that we are now able to look beyond the tumor tissue, which can be very difficult to obtain, and sometimes non-visible or inaccessible. So liquid biopsy, where we are able to look at the circulating cell-free DNA for tumor-derived mutation, really provided a very useful avenue, and we did spend quite a bit of effort on developing digital PCR assays, and then followed by the NGS based approach for liquid biopsy for solid tumors. At the same time, we also had to come up with a framework so that the liquid biopsy oftentimes means just sequential monitoring to many people, which is not really the case. So we established evidence-based examples and utilization criteria for when we perform the liquid biopsy when there is no tumor tissue available at baseline. Sometimes in cases of lung and melanoma, colorectal cancer, I apologize, and melanoma, we can look at the tumor response. For lung cancers, we can look for resistance. You can see there are different time points for this and different clinical applications that have been established. What it meant is that now we are looking at different compartments, sometimes in tumor tissue, sometimes in blood, sometimes in plasma, and that does create this type of a combination of where is the variant coming from, and what does it mean. Is it a somatic variant? Is it a germline variant? Is it a hematopoietic origin chip variant? So this really creates a lot of important questions, but also very important opportunities to bring back to the patient care. So again, you can follow along some of these designations here, and we are moving more and more towards testing tumor, normal, and plasma as much as possible, all three compartments for any given patient, so that we can designate where these variants are coming from. And so that really marked our transition into the next phase, which is we are continuing to grow, but there are some platforms which are reaching maturation. And again, the drivers here are immuno-oncology. In solid tumor, more and more targeted therapies are now leading to more and more treatment resistance. The liquid biopsy success is also leading to the use of liquid biopsy for minimal residual disease monitoring. And similarly, on the hematologic neoplasm side, we have yet another revised neoplasm classification, actually two now, to deal with. So those are again driving the growth. And again, on the NGS platform side, we now actually have multiple configurations available to us, from low throughput to very high throughput. Also, chemistry-wise, we now have UMIs and more sophisticated error correction mechanisms. So this is what is defining the current phase, where we are constantly adding these different sequencers to our portfolios. When we started with MiSeq, now we have a MiSeq and an NextSeq and a NovaSeq, and different flow cells on each of them. Same thing on the ion torrent side, PGM and proton, S5. Now we recently added some GeneXus to look at the rapid turnaround and some other possible applications. So this portfolio keeps growing, and so do our panels. So just showing an example of a 610 gene solid tumor panel, where we are able to perform a comprehensive assessment including MSI, tumor mutation burden, limited DNA based fusion. So more applications are coming out along with more targets. Here's an example of how the MSI calls on NGS based panel that we have perform in relation to the conventional size based or fragment analysis based MSI calls. You can see a very high degree of concurrence when it comes to MSI high and MSI stable status. So these are becoming routine, and we are systematically able to now look at all the tumor types for these important biomarkers. Technical advances rather in chemistry, such as unique molecular identifiers, is allowing us to go deeper and identify these rare variants like a needle in a haystack with higher accuracy. So this is an example of a lung cancer patient. The plasma showed with very characteristic exon 20 mutation and only 3 reads, but with a decent family size, and we were able to identify these, and later on this was also confirmed on the tissue. So again, we are seeing some significant progress and growth in looking broadly but also looking deeply and also looking in for these very rare variants with a high reliability. So this is our overall portfolio, both for solid tumor side, liquid biopsy and hematologic panels, and this is the journey that continues on and on. We are working on some single gene assays here. We have added somatic hypermutation. So again, the need-based assessment and then whatever is the best suited analysis. One important consideration here is that as the panels get bigger, we do need to introduce a normal testing. Otherwise, the variants such as BRCA1 in this case and this other variant, which were actually germline, they would otherwise appear in the tumor-only approach and may confuse the situation. So this is the approach we have taken. We do on all our solid tumor panels, we do tumor normal analysis using blood or normal tissue. What it does is it leads to identification of these potentially actionable germline findings, and we have established elaborate workflows to again handle these in conjunction with our clinical colleagues. Again, some more papers that we published showing these clinical workflows and how we are able to identify these variants and timely manner alert the clinical teams and contribute to the patient care. Obviously, when the panels get bigger, we reach the limitation of how much knowledge we can retain, as well as how much advances in the therapeutics we can keep up with. And so we did develop a system called Precision Oncology Decision Support System in partnership with our Institute for Cancer Investigational Cancer Therapeutics Group, where we are able to look at different variants and identify suitable targets and therapeutic targets, guidelines, FDA guidelines, and so on and so forth. So this is again a very important component. Unfortunately, I won't be able to go through some of these in the interest of time, but just wanted to point out that all of this growth is in fact in a very larger umbrella of a bigger genomics program, where we are able to leverage all of this information and contribute back to the patient care directly. Showing some examples of how these complex platforms are carefully evaluated and placed in production using a large number of QA/QC steps that are carefully implemented, monitored, and optimized. Just showing here the number of QA/QC steps that are performed on different workflows, and this number is actually even bigger now. We've also implemented the same thing at the informatics level. All the samples, they get a detailed QC for run level QC, it's a sample level QC, and also we've introduced a number of unique SNPs in each panel, so that any sample contamination can be detected using this type of a sample correlation matrix. Again, very important measures required for patient safety and quality as we grow in complexity and in size. So in conclusion, the NGS applications in oncology, we think we have just scratched the surface. There are so many different applications that can be performed, and so many different types of readouts that can be generated from these. And so right now we feel like we have just scratched the surface on some of these, and we have a lot more work to do. A lot more platforms are also coming out, and a lot more applications are rapidly making their way from being research applications to the clinical application in prime time. So lessons learned, it's a multidisciplinary effort. We do require a robust pre-analytical process. Extensive validation is the key. We do need to be incremental about replacing the existing workflows. All of a sudden changing everything may result into some unexpected consequences. We do require pathologists as well as highly trained scientists who can perform this and maintain high quality. And also the customization of panel content may increase its utility, and communication with our clinical colleagues is a must, so that we can realize the most value from this entire effort. So with that, I would like to thank again the committee for allowing me to join this wonderful occasion, and I will stop here and we'll take any questions. Thank you.